Hello and welcome back to another lecture. Today we're talking about membrane transport. But before we start talking about that, let's just recap what we discussed in the last lecture. So last time we talked about translation. We said that translation requires ribosomes. We also said that for translation to happen, we need mRNA, which is translated. We need tRNA, and then we need the tRNA synthetases, the ones that make the specific tRNAs. We need initiation, elongation, and release factors, and of all of those are specific types of proteins that do the job. We also said that mRNA sequence provides the information which tRNA is allowed to bind. We said that the ribosome brings the tRNA amino acid complex and the mRNA together, and then facilitates the formation of peptide bonds between the amino acids so that the peptide can start to grow. We said that repeated cycle of tRNA, mRNA binding on the ribosome then creates that longer chain, that the DNA code is usually depicted as a lookup table that translates the codon on the mRNA into the correct amino acid. So you can take that table, you can check what codon translates into what amino acid. And we said that multiple codons can code for the same amino acid, and that was what's called a degenerate code. So those are things to remember from the previous lecture. So today we will talk about membrane transport. So think back to one of the earlier lectures where we discussed biomolecules, specifically fats. And when we talked about fats, we said there was one type of fat that's not really a fat anymore because it's changed around a little bit, but it looks very similar to one in several respects. And that was the phospholipids. So phospholipids and fats are similar in that they have a glycerol backbone and that they have fatty acid chains. But instead of having 3 fatty acid chains, which fats do, phospholipids have that 3rd fatty acid be replaced by the phosphate group and then a small charged head group. And that could be choline, for example. Here's an example for one of those small charged head groups. Which in this case is choline. But they all have this phosphate group that binds that small charged head group to the glycerol backbone. So because of that, we have charged parts in the hydrophilic head, and then we have uncharged nonpolar tails, which of course are the fatty acids. And so you see in this figure here that the top part is the hydrophilic and the bottom part is the hydrophobic part of the molecule. And because of this nature, having both a hydrophilic and a hydrophobic part, that can allow the formation of a membrane. And now the question is, how does it allow the formation of a membrane? Well, if you have a bilayer membrane where you have a phospholipid on the top and on the bottom, or on the outside and the inside, if it becomes a cell, then this lipid bilayer can form a barrier between an outside and an inside where it can mix with water on both sides, inside and outside, but have a impermeable or only semi-permeable layer in between, and that is made of these non-polar tails. So in other words, semi-permeability allows the selective uptake or discharge of molecules into the cell because they can't just simply diffuse in and out. Lipid bilayers can form spontaneously. So if you pour that in if you pour the molecules into a liquid, they can form a lipid bilayer, and they have both polar and non-polar characteristics, and that's called an amphipathic nature. So something that's amphipathic means it's has both polar and non-polar characteristics. So if we take a look more closely here at the different types of fatty acids that you can have within a membrane, then you can also see that these membranes can be made up of different components, and that these different components can have slightly different functions. So saturated versus unsaturated fatty acids we discussed early on. And the reason why one was saturated versus unsaturated was because we have this is unsaturated here because there is still room for more hydrogens. So you could add more hydrogens here. And if you did, then this kink in the structure would straighten out and it would look like this. And that is, of course, because carbons usually form these tetrahedral structures where you have 4 molecules in a 3-dimensional space that can be placed around a carbon. But if you only have 1 hydrogen here, then that becomes no longer tetrahedral, so it doesn't look like a pyramid anymore, if you will, but it looks flat. And so when you have the pyramid type structure interrupted by a flat region, then that introduces the kink in the structure. So if you have those, if you have membranes with mostly saturated fatty acids, they are more rigid than those with more unsaturated fatty acids because there's more space here in between, right? So these can pack really tightly, but these cannot. So unsaturated fatty acids have 1 or more double bonds that introduce kinks in the phospholipid, reducing the tightness of packing. Okay, so far so good. Membranes in cells are not just lipid bilayers. They have other components inside them. And one component that animal cells frequently have is cholesterol. So that is where cholesterol comes from in the diet, from animal cells. So cholesterol is also an amphipathic molecule, so one that has both polar and nonpolar regions. And the reason for having that, or the function that it has, is that it keeps animal membranes from becoming too rigid or too fluid when the temperature changes. And of course, an animal might be exposed to different temperatures during the day or during the year. And cholesterol helps in basically in preventing that the lipid membrane becomes so rigid that nothing can pass through anymore, or that it becomes so fluid that it basically melts. So at warm temperatures, the interaction of the hydrophobic part with fatty acids via van der Waals forces keeps the membrane from becoming too fluid. So you can see that it can sort of intercalate here, and it can react here between this part and this part of the cholesterol. And at cold temperatures, the hydrophilic head group, this part here, the hydroxyl group, that repels neighboring phospholipids just enough so that they don't get too tightly packed. So depending on the temperature, this will come into play or this will come into play, the part of the cholesterol. So that keeps it just at the right level of fluidity, regardless of what the temperature is. All right, so cholesterol is part of the membrane. We said that these phospholipids were part of the membrane. There are other membrane components, and those are often proteins. Many proteins are found in membranes. So membranes can contain both what's called integral and peripheral proteins, and they're embedded or connected to the membrane. An integral membrane is one that spans maybe through the entire membrane and sticks out at both ends. Could just be inside the membrane and just sort of barely stick out. But it's one that is embedded into the lipid part of the membrane. The ones that are peripheral are just sort of on the top. They're not really going through the membrane. But the reason why they're still called membrane proteins is because they're anchored to integral membranes. So they're somehow stuck onto the membrane. They don't float around in the cytoplasm. They are part of the membrane, but they're anchored to it. They're not all the way going through the membrane, such as integral proteins do. So if you think about these proteins, if they have to go through the phospholipid bilayer, then they must have hydrophobic regions within the protein that allows them to do that. And if you think back to the amino acids, when we talked about those and we said there's all these different types of amino acids that have different rest groups, some of which are polar and some of which are non-polar, then of course the ones that are polar are not so easily threaded through a membrane with a lot of non-polar and very hydrophobic molecules right next to that, right? So the membrane proteins can vary in their hydrophobicity or hydrophilicity. So they might have their bits that stick out be hydrophilic with amino acid residues that are polar, and amino acid residues that are non-polar in the hydrophobic region. And then these membrane proteins have a variety of different functions. So I just told you what kind of properties they must have so that they can be embedded or anchored to the protein, to the membrane. But they have all kinds of different functions. And so we will talk about transporters later on in this lecture. So they can allow the uptake of certain substances from the outside or the discharge of substances from the inside to the outside. So they can transport substances in both directions. And that's important because what we also discussed earlier was that certain structures, certain molecules, due to their chemical structure, just have a really hard time just simply getting through the membrane because they're either so large or because they're so charged that they couldn't pass through the membrane. And that is when you have transporters that are allowing the movement from one side to the other. We'll discuss those in more detail. There are also receptors. So those receptors, they can sense that there's something going on the outside. So in this case, this is a molecule that simply binds to the receptor, and then the receptor signals something to the inside. So it relays a signal from the outside to the inside. But this molecule itself does not pass through. So that's a receptor molecule. Then there are enzyme molecules that are embedded in the membrane that are acting as catalysts, as proteins that do something, but they don't float around in the cytoplasm. They're stuck here on the in the membrane, and that's where their function occurs. And then we have anchor proteins that have functions where they physically interact with the outside for attachment. So this is some sort of a structure that this protein anchors itself to, so that it could, for example, anchor to another cell that is on the outside that also binds to the structure. So physical interaction to the outside for attachment. Now let me show you a nice little animation. This is from your book that tells you about the membranes.
The plasma membrane is composed of a phospholipid bilayer, other forms of lipids such as cholesterol, as well as proteins and associated sugars. In this way, the plasma membrane is a mosaic made up of many different components. In addition, the plasma membrane is not static, but instead fluid and dynamic. For example, lipids and proteins move easily in the plane of the membrane. However, movement from one side of the bilayer to the other is less common because proteins and the polar head groups of lipids do not pass readily through the hydrophobic interior of the bilayer. Because the membrane is fluid and made up of many different kinds of components, we use a fluid mosaic model to describe the behavior of molecules in cell membranes. Here we see the fluid membrane in action. A fluorescent molecule is attached to proteins embedded in the cell membrane. The result is that the cell membrane is uniformly fluorescent. A laser is then used to bleach a small area of the membrane, leaving a small area of non-fluorescence. Over time, the bleached area becomes fluorescent as fluorescent proteins move from the unbleached area into the bleached area.
So that last part of that animation showed you an experiment that was done with which scientists found out that there is in fact this fluidity, because the molecules that were destroyed, she said, bleached, they were dissipating, right? And so if the membrane was static, then that wouldn't have happened. That bleached part, that little lighter area that the laser burned into the membrane or on top of the membrane, bleaching those proteins that they had experimentally embedded into the membrane, that would have stayed in one spot, but it moved, and so therefore that was evidence that there must be a fluidity to the membrane. Okay, so we talked about membranes. Now the question is, what's the implications that we have membranes that allow uptake of certain solutes but do not allow uptake of other solutes? And then how can water affect potentially, can be affected by what's inside versus what's outside of the membrane? So that is, there are two principles that we'll discuss. We'll discuss the principles of diffusion and of osmosis. So you may have heard about those terms before. Let's go over them and see what they mean. So membranes allow the active maintenance of a constant state, which is known as homeostasis. And there are two properties that will allow membranes to do that. One is diffusion, and then a few slides down, this will change to osmosis. So let's talk about diffusion first. So if you have a membrane that's permeable, so that means it's a membrane that allows the passage of this green molecule here as well as water, then if you start on this side with a higher solute concentration and a lower solute concentration on this side, where solute means that it's something that's dissolved, right? It's a molecule that is dissolved. It could be an uncharged molecule, it could be an ion. So something that is dissolved is called a solute. So you have a higher concentration here and you have a lower concentration here. There's a net movement of solute from the area of the higher solute concentration to the area of the lower solute concentration. So in other words, concentration gradient is always important. If you are trying to assess the movement of these membranes, it always goes from higher concentration to lower concentration. And this is now before the equilibrium. So this is, let's say, the start. We just filled this beaker that has this membrane in between with these two different solutions to the same level, but this one doesn't have as many solutes in it as this does. And now the molecules can move over. That is going on until there's no more net movement, because the solute concentration is the same on both sides. Now it says no more net movement because the diffusion continues to occur. So there's still movement of water molecules back and forth and of these solute molecules back and forth, but at the same concentration. So there's no net movement anymore. The concentration will now stay the same at the point where the concentration is the same on both sides. So that's what you have here in diffusion. So what kinds of diffusions can occur along a membrane? Well, diffusion can occur through a membrane either through, it's called simple diffusion. Simple diffusion then means that it just passes straight through the membrane. Or if that cannot happen because the molecules are too large or too charged, then they have to go through a protein. And so there are different types of proteins. There are so-called carrier proteins, and there are channels. Carrier proteins are a little bit more complicated. They require this change in conformation. They allow the uptake. They basically bind this molecule and then spit it out on the other side. But there has to be change within the conformation, the shape of the protein. While the channels they can open and close, but they basically just let stuff through. And so if you use channels or carriers, then that is called facilitated diffusion, because there's still only, this is still only going to work if there's a higher concentration of molecule on the outside than inside. So this still goes along the concentration gradient, where only then will you have a movement from the outside to the inside. So only if you have a concentration gradient difference. Again, the solute only moves along its solute concentration. But what these proteins do, they allow the passage that otherwise would be restricted by the lipid bilayer. So the membrane does not restrict, so diffusion can occur if the membrane does not restrict diffusion due to size or charge. It can occur if it is aided by a transporter in facilitated diffusion, and if the concentration of the diffusing molecule is greater in one area versus another area. Okay, so these are still examples of diffusion. So now let's talk a little bit about osmosis. So osmosis can also lead to some sort of homeostasis, a constant state. But in this case, in this example, we have again a selective permeable membrane, and we have the membrane in between two compartments, and we have higher solute concentration on one side and lower solute concentration on the other side. And the book says there's also lower water concentration here and higher water concentration here, and that's a little bit difficult to grasp, right? Because it's just water to the same level. But there's a tiny little bit more water here, because here, if it's to the same level, then the presence of all these molecules in there, they also take up some room, right? And so therefore, if it goes to the same level, then it must mean that there's a little bit less water in here, because otherwise, if you added a bunch of stuff, then it would rise, but it doesn't rise, and that's because there's less water in there than over here. And so that's why they're called this higher water concentration and this lower water concentration. So what happens over time now is that the membrane allows the passage of water, but it doesn't allow the passage of solute. So this would be an example where the solute does not pass through the membrane. So there is no way for this particular membrane, and remember, membranes are different. They can have different types of proteins inside. They can have different properties. If this is a membrane that has a property that will not allow this particular solute to go through, then it still allows water to go through. So now water will pass from the right to the left, and it will change its level until it reaches the same concentration of these solute molecules per volume. So concentration always means number of molecules per volume. And so here you have 3 molecules per this volume, so there should be twice as many molecules per this volume. So this is not directly to scale, but that's what is meant here, that you have the same concentration of water now and solute per water, because it's now diluted to the same extent. I have another slide here that shows that. So here you have water that moves through this example, but we have a sugar molecule as an example that cannot pass through it, right? So that this just shows you that water is small enough to pass through this membrane. And then just to sort of sum up what I said, if a membrane restricts the flow of a dissolved molecule, then water will flow through the membrane to dilute the more concentrated area. So it moves into here, which is more concentrated. If the membrane does not restrict solute flow, then the solute will move from higher concentration compartment to the lower concentration compartment until equilibrium is reached. But if it does restrict solute flow, then that is not happening, and the change of the water level is what happens. So here's another way to look at this. Here you have higher concentration of one solute, lower concentration of solute. You do have water permeability, so water can move through here, and water will move through here because there's a higher concentration, there's a lower concentration here, but these molecules are too large to pass through, right? That was the example where the sugar molecules cannot pass through, but the water molecules can go in both directions, but they will always go towards the higher concentrated area, and therefore the water level in this U tube here rises. So the rule of thumb that you should remember from this is that water moves towards the compartment with a higher solute concentration. We will do some experiments in lab where you will see that happening, but you should remember that water always moves towards the compartment with a higher solute concentration. So what you will see in lab is an experiment where we will use blood cells and we will put them in solutions that have different concentrations of salt or of solutes. So this particular solute that we will use is salt, just regular table salt. And so you can describe the difference of concentration of solutes in 2 compartments, let's say inside versus outside, by these terms here. And so these terms are terms that you will encounter. And so we need to discuss what they mean. So there's a hypertonic state, an isotonic state, and a hypotonic state. A hypertonic solution versus the inside, so solution is the outside, means that the solute concentration is greater on the outside than the inside. So it's hypertonic. Hyper means high, strong, much more than the other, hypertonic. Then you have isotonic. That just means iso meaning the same. So it's an isotonic solution. It's the same concentration of solutes on the outside and the inside. And then the opposite of the hypertonic is the hypotonic, hypo meaning under or lower, where the solute concentration inside the cell is higher than the outside. And note that we talk about a hypotonic solution. So we are specifying what part is hypotonic. Theoretically, you could also talk about the state of the inside, right? So if we were say, well, what's the inside? That could also be hypo, or hypotonic to something that you compare it with. So you cannot simply look at a jar of something and say, oh, that is a hypertonic solution. It's always in relation to something else, more than this other compartment. So hypertonic solution is always in relation to the inside of the cell that's inside the solution or another compartment that's next to the solution. You cannot just have one solution with no reference point that is any of these three states. So when you have that, then because of osmosis and because of that, the properties that I just explained with just the jars and these U-tubes, of course, also act in nature. And so what happens is that water always tries to dissolve, to dilute the more concentrated area. And if that is more concentrated on the outside, then water will move out of the cell, and therefore the cell will shrink. Right? Water will move out. It will try to dissolve, to dilute the outside. Water will try to dilute the outside, which is higher concentrated. Might be futile because it's so much more concentrated, but it will still do that. And so therefore, the volume inside will shrink because all the water goes out and nothing else moves in to replace that. In a normal cell, typical looking cell, if you put that in an isotonic solution, then water will move in and out. There will be no loss or gain of water. And so if you if you want an isotonic state between a cell and its blood, for example, so blood cells inside the blood, then you want to be sure that the blood is not too high in salts and not too low in salts. And so that's why when you have isotonic drinks for sport drinks, for example, they will provide a level of electrolytes, it's usually called, that's essentially dissolved salts and sugars, that will have the same concentration as what is inside a cell. So that's isotonic sport drinks. They have the same concentration as inside the cells. And here the isotonic solution would be inside and outside, same concentrations. Now, if it's reversed and you put these cells that have some salts inside into hypotonic solutions, distilled water that's completely clear of all kinds of salts in there, then suddenly the concentration is the other way around, right? So there's now more salt in the inside, and so therefore water will try, like it always does, to dilute this until it's the same concentration. That is also sort of futile because if this is completely devoid of any kind of salt, it will continue to go in there, and it can go into a membrane so much that can actually rupture that cell. So that means it's lysed, it's broken, it's broken open. That can happen in very hypotonic solutions where the membrane is not strong enough to withstand the pressure of the water coming in. So in plants, however, that's slightly different because plants have, in addition to the membrane, they also have a membrane, they have a cell wall, and the cell wall is rigid and allows, just like a balloon that you blow up, right, that can burst at some point. But if you put that balloon in a box, in a shoebox, and the balloon is big enough that it won't burst inside the shoebox, now that you pump up the balloon, it will just go as far as the shoebox will allow it to go, and after that it will restrict the amount of inflation, and so therefore will prevent the balloon from bursting. And that's the same principle that you have in a plant cell. So here we have the state where water moves in because we have the hypotonic solution, so more salts on the inside of the cell and less on the outside. Water moving in, but it cannot, it gets more turgid, more inflated, but it won't burst because of the membrane, because of the cell wall that is preventing the membrane from doing that. In an isotonic solution, water moves in, water moves out, so the cell is happy. It's not getting very swollen. It's not very it doesn't become too flaccid. But then in the case of the hypertonic solution, water moves out of the cell. And what you will see there in the microscope when you look at that, and we will do that in lab, you will see that the membrane, these are the membranes, right? So this is one membrane, the outside of the cell. You see these strings, they are attached to the membrane. And at some point when you do this for too long, they will actually come off here, and you will just see a tiny little circle inside, and that circle inside will just be the so-called plasmolysed cell that's inside the cell wall now. So you can see that when you put it in a hypertonic solution, like a strong salt solution. All right, I want to show you this in an animation as well. Sometimes moving things can be easier to understand than just static things I can show you on the slide.
Osmosis is the movement of water molecules from regions of high water concentration to regions of low water concentration. Osmosis can be observed using a beaker and a semi-permeable membrane that allows the passage of water, but not the passage of dissolved solutes. On one side of the membrane there is a high solute concentration, and therefore low water concentration, and on the other side there is a low solute concentration, and therefore high water concentration. Water moves from regions of high water concentration to regions of low water concentration. Water moves down its concentration gradient until the water and solute concentrations are the same on both sides of the membrane. Osmosis can also be seen by watching what happens to red blood cells placed in solutions with different salt concentrations. When a red blood cell is placed in a high salt or hypertonic solution, water moves from the inside of the cell to the outside of the cell by osmosis to equalize the water concentration on the two sides of the cell membrane. As a result of this water movement from the inside to outside, the cell shrinks. Conversely, when a red blood cell is placed in a low salt or hypotonic solution, water moves from outside the cell to the inside of the cell. This time the cell swells or even bursts open.
All right, so that principle of water moving in and out, that's exactly what we will look at in lab, and we will use red blood cells for you to experiment with and see under what conditions you will have lysis, bursting open of these cells, and at what point the concentration of salts on the outside might equalize the concentration of salts inside. All right, okay, so now we'll come back to the membranes, and we'll come back to the proteins that are inside these membranes. And what I want to tell you about now is that there is also the option for cells to pick up things against their gradient, their concentration gradient. So up to now, I've told you, and I said, remember this as a rule of thumb, or remember this as one important principle, that solutes and water will always move from the higher concentration to the lower concentration. So under what conditions can substances be, let's say, imported into a cell against their concentration gradient? So imagine that you have a cell and it wants to pick up sugar because it wants to break down the sugar as a molecule to give it energy. Well, after a meal, there might be a lot of sugar on the outside and some sugar on the inside. And so therefore you may have a lot of movement just through its concentration gradient. But if there is now a reasonable amount of sugar inside and there's now less on the outside, there's still some there, and the cell would still like to use that rather than just let it pass by and say, sorry, we're full. Concentration gradient is currently greater here than on the outside, so therefore nothing can come in. So it would be great to utilize all of that, and that is when the concentration gradient gets circumvented by what's called active transport. And that costs a little bit of energy. And I want to show you how that works. So in primary active transport, solutes can be moved against their concentration gradient by the use of energy. Here you see the concentration gradient in for the example of sodium, the purple molecules here, ions, and potassium, the pink ones that are more concentrated here on this side. And so the concentration gradient is greater on the inside than on the outside. For sodium, it's greater on the outside than for the inside. So we have an example where cells usually need more potassium than sodium. And so they want to get rid of sodium, they want to bring in potassium at the same time. And there is a membrane protein that can actually do both things. So it can pick up sodium on the outside it can pick up sodium here, here on the inside and bring it to the outside. So it loads the membrane protein, and then after the breakdown of ATP, which is the energy currency, which is a molecule that's high in energy, and we'll talk about that in much more detail later on. So for right now, it's good enough to just know that it's a molecule and it contains energy. And when that molecule is broken, it releases the energy, and that energy is then used for the conformation change. You see that the protein changes its shape. That's called its conformation. And so the sodium ions are spit out on the other side. And at the same time that the conformation is such that the sodium ions can be spit out, they can also pick up the potassium ions basically on their way back. So it's sort of like a revolving door, except that you can only have one kind go out and one other kind go in. But it goes in through the same revolving door, through the same membrane protein. And so then potassium is moved in. So that goes against the gradient, but it costs some energy. So transport protein picks up sodium, uses energy in the form of ATP, pumps out the sodium, the same protein then picks up the potassium and then delivers it to the cell. So that's transport where you have that one protein that's directly coupled to the use of energy. There's also a way how energy can be stored, if you will, and almost like a battery, be used for the powering of other molecules. Let me explain what I mean. That is called secondary transport, a secondary active transport, because it requires active use of energy. So in secondary versus primary, what we looked at in the previous slide, active transport, what happens is that first this battery, let's call it, gets charged. And the way it gets charged is that protons, they make up the battery. They are pumped across the membrane via primary active transport. So you have a molecule that, just like in the past slides, uses ATP and then pumps these protons, so hydrogen ions, pumps them out of the cell and into the extracellular space. So now you have a concentration gradient where you have more protons on the outside than on the inside. And you have also a charge gradient, because these protons were all positively charged, right? So the proton pump generates an electrochemical gradient, as it's called, with a higher concentration of protons outside the cell and a lower concentration of protons inside the cell. And then in this case, you have a molecule that is called an antiporter, because it brings one thing in and another thing out at the same time. It uses this proton electrochemical gradient to move a different molecule out of the cell against its concentration gradient, or in or out. And so in this case, this molecule goes out at the same time as the protons flow back. But these are different molecules, right? So it's not the same protein that uses ATP and then delivers this particular molecule in one direction or another. And that would be primary. But here it just uses the gradient, the proton gradient that was built up by another protein. And that's why it's called secondary. So secondary means it uses the gradient that was built up by somebody else that used the ATP. This guy doesn't use ATP, and so therefore that's why it's called secondary active transport. It's still active transport, meaning it cost ATP, but it didn't cost this guy any ATP. It cost this other protein ATP. So you can use one proton pump and make a bunch of move a bunch of these protons out, create this gradient, and then you would only need, you could verify the types of proteins that then specify for using a proton and deliver or take up a different molecule. But it would always be protons that are doing the trick, and so therefore that's maybe a little easier for the cell to create these, because they don't have to be all completely different. They use the same overall mechanism, the hydrogen electrochemical gradient, or the proton concentration gradient, as the means of pumping. So they only need one proton pump and then have a variety of different proteins that use the proton gradient. So that is called secondary secondary transport. So I wrote here on the side, primary versus secondary. Primary uses ATP directly to power a reaction. Secondary transport powers various different types of reactions using the electrochemical gradient, which was originally built with the ATP. So for that, I also have a little animation. Let me show you that.
Some molecules are actively taken up or removed by cells against a concentration gradient. In these cases, energy is required. For example, the sodium-potassium pump is a transmembrane protein that moves sodium ions out of the cell and potassium ions into the cell. The sodium-potassium pump uses the energy of ATP to pump 3 sodium ions out for every 2 potassium ions in. As a result of the action of the pump, the concentration of sodium ions is high on the outside of the cell relative to the inside of the cell, and the concentration of potassium ions is high on the inside of the cell relative to the outside of the cell. Because this pump uses the energy of ATP directly to move molecules in and out, this process is called primary active transport. In some cases, the energy of ATP is used indirectly. For example, some transport proteins take advantage of gradients set up by primary active transport. In the example seen here, a transmembrane pump uses the energy of ATP to move protons from the inside to the outside of cells, creating a concentration gradient of protons. In addition, the movement of protons results in a charge difference between the inside and outside of the cell. The concentration and charge difference together is called an electrochemical gradient. Such a gradient stores energy that can be harnessed to move other molecules in or out of cells against their own concentration gradient. Another transmembrane protein then couples the movement of protons back into the cell down its electrochemical gradient with the movement of another molecule out of the cell against its concentration gradient. This process is called secondary active transport.
So in secondary active transport, the coupling of the gradient, that is what gives it the energy, right? So it powers this molecule because there's a gradient, so the protons, they do move along the gradient, and that is what powers it. But at the same time, they sneak this molecule out because the protein here, this membrane protein, is shaped in such a way that it can swoop up one of these molecules. It fits just in, but only when there is the push that comes basically from the protons that are moving through the membrane protein. All right, so let's just sum up what we got here today. We said that membranes are semi-permeable, fluid, and they contain proteins. We said that cholesterol allows the membrane to stay fluid enough, but not too fluid in changing temperatures. And those are just in animals. Those are animal cells that have cholesterol. Diffusion occurs across a membrane along the concentration gradient of the solute in question. So if you're thinking about one solute and you're looking to see where it moves, then it will always diffuse across a membrane along its concentration gradient, from higher to lower. Solutes move from higher to lower concentration. Water also moves from higher to lower concentration, right? Remember that when there's more water per volume, because there's stuff inside dissolved solutes, that is how you can have more water, higher water concentration per volume. Membranes can restrict solute passage, but membrane proteins can facilitate passage of specific molecules. So these proteins are specific for just maybe one kind of molecule. They won't let just anything through. They are specific for one kind. Membrane proteins can allow passive diffusion, that's called facilitated diffusion, or active transport. So if I will ask you what's the difference between passive and active transport, then one is simply passive diffusion, or it could be facilitated by membrane proteins. And then if a membrane restricts solute passage, water will move through the membrane to equilibrate the concentration difference, and that's osmosis. And we talked about that in the context of water moving in and out of cells to the point where they can even lyse, where they can even burst the cell. And we discussed the difference between plant cells and animal cells there because of the cell wall. All right, and that's it for today. I'll see you in class.